Alright guys, these are the questions you received today. You probably want to go over these before your test on Wednesday. So this question is asked about DNA replicates. And if you remember, okay, DNA can only be replicated in the 5' to 3' direction. 5' to 3' direction. Okay, that's DNA and RNA. So they gave us our original DNA that's been unzipped by DNA helicase. That's right, DNA helicase. And so if this is the 5' end, this is going to be we're going to be making the opposite, which is the 3' end. Okay, and this is the 5' end. This is 3 here, so this is 5, and this is 3. DNA is made from 5 to 3, okay, but DNA is not made from 3 to 5. So A is incorrect, even though it is made of fragments. On B, this is 5, this is 3 again to 5, and it is not going to be copied 5 to 3. See the arrow? D is the same thing. We got a 5 to 3. That's a 5, so this is 3, and this is 5. Okay? DNA cannot be made 3 to 5. Looking at this one, C here, we got a 3' to 5', so the new strand is 5 to 3, and this is our leading strand. So that is correct. Okay, both C and D, or both C, A, and B, this strand is correct. Okay, it's this lagging, the lagging strand that was incorrect. If this is 5', this is 3'. Okay, 3', and this is 5'. And we're going to copy from 5 to 3. We're going to do it in these small little fragments. We call those Okazaki fragments, right? So here we have our fragmented lagging strand and our continuous leading strand. So C is the correct answer on the first one there. The next one, let me write this for you so you can see it. So this is talking about lactose digestion in E. coli up here at the top. Okay, and it's talking about hydrolyzing an enzyme. Okay? They have this picture here. There's RNA polymerase. There's our repressor protein. Make sure you know what our repressor protein is. And here's lactose. The question asks, which of the following figures correctly depicts the interactions of the lac operon, okay, when lactose is not being utilized? That means this guy is not present. So A shows the lactose is bonded to the repressor protein and it's released. That is incorrect because lactose is not being utilized. On B, again, the lactose is bonded to the operator. That is incorrect. On part C, okay, we have RNA polymerase copying away, okay, but lactose isn't present. So C is incorrect. And on D, we have the repressor protein is present, no lactose is present, so therefore we can't copy the gene. So D is correct answer. Again, it's because knowing that for the lac operon directly, lactose has to be present. Okay? If lactose was present, then A would be the best answer choice. But it said lactose is not being utilized, so D is the correct answer choice. Okay? All right, very good. That's the first two. Let's look on the back now. All right, on this one, you look at the picture first. We have a phage, which is a bacteriophage, and it's got DNA, and here's our RNA, or here's our DNA and our bacteria. It's an R plus and an R plus, okay? And then here looks like the DNA's been in there, and it looks like we're making lots and lots of copies of our DNA, and now we've made tons and tons of baby viruses, and they're lysing or rupturing the cell and escaping. Here, one of them is attached to another cell, but this cell is a little different. It's R minus S minus. Okay, so it's got some different genes here, and it's adding an S plus piece of DNA here. Okay, and the new bacteria, okay, you'll notice is not copying DNA and it's not being taken over by virus. It looks like it's R minus S plus. I mean, it looks like we added a gene there. We added S plus. Okay? So So let's look at our 4 answer choices there. Okay. The first one says bacterial protein transferred from the donor bacterium by phage to the recipient. Bacterial proteins transferred. Okay. No, we transferred DNA. So A is incorrect. The recipient bacteria incorporates the transduced genetic material coding for phage proteins into the chromosome and synthesizes the corresponding proteins. That's what's happening here. Okay? We don't see it at the very end, so B is incorrect. The phage infection of the recipient bacteria and the introduction of DNA carried by the phage cause increased random point mutation of the bacterial chromosome. Okay? We're not seeing any mutation here. We're seeing R minus, okay, and S minus becoming R minus and S plus. So that is incorrect as well. And D, the DNA of the recipient bacterial chromosome undergoes recombination with DNA introduced by the phage from the donor bacterium, leading to a change in the recipient's genome. We're going from S minus to S plus. Okay, so D is the best answer choice for question number 3 there. Okay, and lastly, so we have two segments. This one's got a lot of A's and T's. This one's got very few A's and T's. The sequence for the 2 short fragments of DNA are shown above. Which of the following is one way in which these 2 segments would differ? Segment 1 would not code for messenger RNA because both strands have a T, a base not found in RNA. Well, if there's a T here, you write an A on RNA. And if there's an A on the DNA, you write a U. So that is incorrect. Faulty thinking there. Segment 1 on B, segment 1 would be more soluble in water than segment 1 because it has more phosphate groups. Remember, phosphate groups is found on all of the nucleotides. Okay? It's part of the molecule, right? Phosphate group, the 5-carbon sugar, and the nitrogen base. So they all have that. So that is an invalid statement as well. Question number 3. Segment 1 would become denatured at higher temperatures than would segment 2 because the AT base pairs have 2 hydrogen bonds, where GC base pairs have 3 hydrogen bonds. Okay? A and T do have 2 hydrogen bonds, and G and C do have 3 hydrogen bonds. Okay? So that is one way that these 2 segments would differ. Okay? So C looks like a good answer choice. Let's look at D to make sure. Segment 1 must be from a prokaryote because it has predominantly AT bases. Okay? The amount of A's and T's does not determine whether it's a bacteria or not. So that is incorrect. So C is the best answer choice. 2 bonds are easier to break than 3 bonds. So C is the correct answer. I hope that you answered these all correctly, and if not, you now understand why your answer was wrong and what the correct answer now should be. Hopefully you studied. If you need additional help, please come by and see me during Academic Advantage tomorrow. I'll see all you guys on Wednesday to take your unit test. See you then.